Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really awesome guest involved in science, technology, medicine, creating a better tomorrow for literally millions of people out there. Today on the show, we have the honor of being joined by Dr. Fred Grossman. He's the president and chief medical officer of a company called Coya Therapeutics. They are a clinical stage company focused on developing multi-modality Treg therapies for various neurodegenerative diseases where they've already developed strong proof of concept data in ALS, as well as some early data in Alzheimer's disease, and then working throughout the autoimmune as well as metabolic disease domains as well. Dr. Grossman brings over 2 decades of drug development experience, having held many senior level executive positions in both large and small pharma companies, leading development of various blockbuster drugs along the way for indications of significant unmet medical need, has had close relationships not just with thought leaders worldwide, but also negotiating drug development and various other projects with the FDA and global health authorities. Dr. Grossman held executive positions at many pharma companies, including Eli Lilly, J&J, Bristol Myers Squibb, and Sunovion. Served as also the president and chief medical officer of Glenmark, the huge generic pharmaceutical company in India, overseeing development of an entire pipeline in generics, as well as complex generic candidates and next generation biologics. Dr. Grossman also previously served as chief medical officer of Mesoblast, developing various allogeneic cell therapies for inflammatory diseases. So he has a lot of expertise also in the cell therapy domain. Dr. Grossman is a board certified clinician in psychiatry, as well as a fellow of the American Psychiatric Association, and was also a fellow at the National Institutes of Health, where he did all sorts of interesting work in pharmacology. And he's held several academic appointments and authored numerous scientific publications along the way. Really honored to have him with us today, and a very exciting set of topics to get into. Dr. Fred Grossman, thank you so much for taking time to come on the show.
Thank you. Glad to be here.
Oh, great to have you. I'm really looking forward to touching on a Nobel Prize winning set of topics, and you're looking forward to getting into T-regs. But before we go there, I would love to, Fred, start off a little bit about you, because, I read a little bit about your background. I took a look at some of the papers you wrote back in the day, sort of even before you got involved in the pharmaceutical industry, studying antipsychotics and mood stabilizers. I saw a really interesting paper in there on anticonvulsants and agitated dementia patients. Take us back a bit. What got you interested in this area in general of psychiatry, and what led you into the pharma industry ultimately?
Well, it's interesting. I actually studied philosophy of science as an undergrad, and using science as a tool has always been intriguing to me, because of people often misinterpret what science is. Science is nothing more than a tool to get closer to the truth. And it was always remarkable to me that people misinterpret what science is. It's not a belief system, it's not a religion, it's not politics, it is merely a tool. And within science, it was always interesting to me to try to understand, what is it that gets you closer to truth? And why can't you ever have definitive answers? And it's because what science tries to do is to create a situation that explains what you're trying to study and gets as close as possible to your primary variable as you can. And it's never definitive. So it just takes me back when I was teaching medical students and residents, and many areas are gray, and sometimes you have to make decisions. But when I would discuss rotations or colleagues, uh, I would always say, if you ever are trying to understand a concept and the physician you're talking to says, in my hands, this is what happens, run for the hills, because it tells me that that physician does not understand science. And start asking that physician questions. Ask them, well, in your hands, who evaluated the patient? Was it you, or
Hmm.
was it an objective evaluator? How many patients did you see? 100? Okay, and you're drawing a conclusion about 100 patients. Did you have an analytic tool to determine whether what you're seeing and your intervention actually did what you thought it was going to do? And so on and so forth. And those are the kinds of questions that were always interesting to me, and why I geared myself towards science and towards understanding what's behind things that are important to men. So obviously, moving into medicine, you're focused on diseases. And
Hmm.
how can you treat those diseases and either cure them or make them livable? And as a physician, you have the opportunity to have a large practice and treat patients and see how you can intervene. But as a scientist, you get to study those, and you get to open up new areas that can open up new therapies that can have a major impact. So when I started, when I was at NIH, I was around some really bright people, and it was fascinating to me to try to understand the pathophysiology behind illness, particularly psychiatric illness. Why would people hallucinate? And what can you do in schizophrenic patients to try to treat them? Why do people get depressed? And all of these are biologic phenomena of the brain. And it wasn't until I had my first experience in the pharmaceutical industry that I saw the complexity and the high science that was done there. And that's what intrigued me and got me started on my career in R&D in the pharmaceutical industry. And I have to tell you that it really pained me. I would see people in groups that are working tirelessly through the weekend, in the evenings, and then I would see articles written comparing the pharmaceutical industry to the cigarette industry.
Hmm.
And it was remarkable, with one group of companies that were harming and others that were curing and saving lives. And the pharmaceutical industry, especially the large pharma companies, are the most democratic institutions, in the sense that there are groups of hundreds of people doing high science trying to get their therapy moving forward, but there's a limited amount of funding. And I've been on the side of trying to get funding and on the side of making the decision of which areas to close down. And it's a tough decision, and you have to rely on basic scientific principles and understand the data and how these programs were de-risked. So it's a long-winded answer, but it's something that has fascinated me from the start until this day with Coya. I'm very fascinated by the process, and we can get into this, I guess, as we move along.
Hmm. Absolutely. And there's one other place I wanted to stop before we get to Koya, because I think it's a very important part of your background story. In addition to obviously extensive work with small molecules, and then when you were in the generics business, you had experience with biosimilars. You, as also mentioned, you've also been in the allogeneic stem cell space with Mesoblast, and in this era, I mean, we're going to be talking about broadly in a little bit about immunoregulation and this ability to modulate, which I think, when we put sort of immuno-oncology over here for a minute, when it comes to a lot of what you know about the sort of the stem cell space nowadays, something they're very good at. And I'd love to you just give a couple minutes of sort of some of your experiences in developing cell therapies, because obviously this plays into the pipeline that we're going to be touching on, and aside from the cool biologics, some of the living cell products as well that you're working on.
That's right. Well, as you noted, small molecules are just that. They're chemicals that you can easily predict and easily understand the pharmacokinetics and pharmacodynamics and the
Yep.
targets that you're hitting. When it comes to cell therapy, it's much more complex because you're dealing, first of all, with manufacturing a biologic, which is very complex in and of itself.
Yep.
And when that's given to a human, it's going through a various biologic systems. And whether it's extruding anti-inflammatory material or not, you have to kind of trace where this cell goes. So does it first start in the lungs and then circulate to the liver and other areas? So in what space is it starting to have a pharmacodynamic effect? And is that pharmacodynamic effect predictable? So I think that stem cell therapy is fascinating and critical. It also leads to CAR T therapy, where you're pinpointing and reprogramming cells to go after specific tumor cells.
Mm-hmm.
But in terms of stem cells, it promotes growth in various areas, but the way we were using it was its anti-inflammatory effect and directing that effect specifically. And I was very happy to see that Mesoblast was able to get approval in graft versus host disease in children, which is a horrendous disease.
Absolutely. Absolutely. It was nice seeing that a couple months ago as well. But yeah, no, I appreciate you laying that groundwork, and I want to sort of for the audience to understand that you've seen all these different types of therapies and all the sort of the interesting dynamics that go on with developing them. So that's great. Yeah, let's go into Coya now, and clearly, you are at the forefront of designing and developing novel therapies, which will touch on both biologic and cell based. And it's interesting. A couple of weeks ago we actually talked about Dr. Sakaguchi on the show and winning the Nobel Prize, and we know that he's an advisor to your company. If everyone listening, watching, you can take a look at that episode a couple weeks ago. But another name we're going to be talking a little more about is Dr. Stanley Appel, who was also on your scientific advisory board, that really did key work over the last couple of decades in sort of understanding not just the connection to between things like ALS and hyperinflammation and hyperimmune response, but did a lot of the early work in understanding how T-regs and T-regulatory cells go wrong. What happens, how hyperinflammatory response leads to all sorts of negative functions on microglia and so forth, but also a lot of sort of the epigenetic alterations and different biology that happens when T-regs go wrong. And I'd love for you if you could take a little time just to introduce Dr. Appel to our audience and a little bit of the key findings, specifically in the area of ALS that he worked on the last many years, because this leads actively into your drug development programs.
So let me say it's a privilege to have Shimon Sakaguchi, who, as you noted, won the Nobel Prize for Medicine on our scientific board for, in fact, discovering regulatory T cells. And it's a privilege to work with Dr. Stanley Appel, who's had decades of research on oxidative stress and inflammation in neurodegenerative disorders. And he's produced a huge body of work. So we're really standing on the shoulders of these giants. So let me step back for a moment and discuss T-regs and why we're focusing on it, and how Dr. Appel has done such elegant work to determine the etiologies of neurodegenerative disorders, including ALS. So regulatory T cells are very important because they maintain a homeostasis of your immune system.
Mm-hmm.
It puts the brakes on excessive activity in the immune system, which is there to produce inflammation, to go after the foreign bodies and infections that occur. And the problem is, if there are no brakes to that system, then that system starts to attack itself, attack the body.
Mm-hmm.
And complexes are formed that are very toxic, that begin to circulate and are released and start to kill, in the case of ALS, destroy motor neurons.
Mm-hmm.
So let's think about regulatory T cells for a moment.
Okay.
What Dr. Appel has done over the years was maintain that that is the basis. Dysfunction of regulatory T cells is the basis of ALS, which is a horrible progressive disease for which there are no adequate treatments. Also, there's an increase in oxidative stress. So people with ALS have this highly inflamed state. Toxic complexes are formed, T cells, cytokines, which invade the motor neuron and kill the motor neuron. So the thinking is, well, what if we can somehow optimize this area, these regulatory T cells, which are at the center of this homeostatic balance of the immune system? So what Dr. Appel and his group have done, which is really very elegant, was to remove those dysfunctional T cells, for example, from ALS patients. And by dysfunctional regulatory T cells, I mean lower numbers of regulatory T cells and lower suppressive function. So the balance is off. So he removed those cells, and his team optimized, meaning increased the number of regulatory T cells so that there were higher numbers and increased suppressive function. And he gave them back to patients with ALS.
Mm-hmm.
And he found something remarkable. So first, in some patients, it inhibited the progression. Now, people with ALS have a predictable progression, meaning they get worse on a scale that's used often, ALS FRS scale. And they go down by anywhere from a half point to a point and a half every month.
Okay.
And when that happens, 1 point could be very significant. It could be the difference between being able to walk and not walk and so on. So when he gave it back to this group of patients, it inhibited that progression. And then something interesting happened. In a few of the patients, it didn't work very well. So not only did the regulatory T cells not maintain its numbers, it didn't maintain its suppressive function either. And that correlated with continued progression. So he thought this through, and the realization occurred that, in fact, these regulatory T cells, you can optimize them. You can make them work as they're supposed to work. But if you put them into an inflammatory environment, they're going to revert back to being dysfunctional.
Mm-hmm.
Dr. Appel calls it the inflammatory milieu. And that was an important finding, because what that led to was, well, maybe the cell therapy isn't the way to go. Maybe there's a way of increasing regulatory T cell number and increasing the suppressive function without having to pull out the cells and putting them back in the inflammatory environment, not have them work very well.
Mm-hmm.
So it's very interesting. Interleukin-2, or IL-2, which is a cytokine that gets circulated in the body. If one is given a high dose of interleukin-2, that increases inflammation. So it revs up the inflammatory system, and that's why high-dose IL-2 in the form of Proleukin is an approved drug for cancers.
Yep.
However, if you give a low dose of interleukin-2, it does a couple of things that are remarkable. It increases regulatory T cells, and it increases the suppressive function. So that's great. This is now a pathway to increase regulatory T cells, which can tamp down inflammation and perhaps stop progression in neurodegenerative diseases. The problem is, when you give the low dose IL-2, and you might see an increase in the regulatory T cells, if you're not tamping down the full inflammatory environment, they're going to revert back to being dysfunctional. And so, in fact, just giving low dose IL-2 is not enough in these patients that have this elevated inflammation. So Dr. Appel and others have done this as well, have gone back and said, well, what else can we add to change the environment to have low dose IL-2 have a sustained effect in people who are suffering with ALS so that the regulatory T cells remain high and the suppressive function continues and the balance is created. And after some trial and error, found that the optimal combination was low dose IL-2 and CTLA-4 ligand, which is abatacept, which is used to treat rheumatoid arthritis. And what's important here is that our immune system is a redundant system. And while it's okay to go after specific targets, I think it's clear now that one needs combination therapies to hit main targets and hit multiple targets. So this combination of low-dose IL-2 and CTLA-4 turned out to be very effective, and I'll explain why.
Okay.
First, Coya has developed a proprietary subcutaneous low-dose IL-2 because it doesn't exist, low-dose IL-2. So and the importance here is that both the Coya 302, which is a combination of low-dose IL-2 and CTLA-4, is in subcutaneous form, the same form that patients who are taking GLP-1 or insulin can self-inject, so they don't have to go into the clinic and get infusions. So what Dr. Appel did was to give this combination to a variety of neurodegenerative disorder patients, but in particular, patients with ALS, and measured using the scale I mentioned before, ALS, FRS, and measured biologic measures to see whether regulatory T cell numbers were high and suppressive function were low, measured whether inflammation went down, and something remarkable was observed. In 4 patients who were given this combination, over a 6-month period, there was little to no progression. Now, I can't tell you how important that is, because over a 6-month period, you have expected on the ALS-FRS scale a reduction of at least 6 or 9 points. And that puts patients into that area where they're becoming dependent on devices to eat, to ambulate, dependent on family members. So the fact that over a 6-month period, there was little to no progression was remarkable, but it was a small number of patients. And it gets back to what I was saying before, that in science, you need to go to the top of the pyramid and have placebo-controlled large studies. And that's where we are now. So in those 4 patients, there was little to no progression. And importantly, biochemically, the regulatory T cells with therapy went up and stayed up. And the suppressive function increased as well. So this combination created exactly what you'd want to see, which is regulatory T cells doing their job, maintaining themselves through measures that are biologic measures as well as clinical correlates. And patients continued up to a year. And there was stability as well after a year. So look, when I saw that, I said, first of all, I have to be part of this company. So, at a time when I was thinking of retiring, this checked all the boxes for something that can be really dramatic and significant. So we took that information and we created a pivotal type trial that just started, that is powered fully. And I'll talk about that in a moment, but we had discussions with the FDA. And a bit about the FDA, because we're sort of in fraught times right now. The FDA is made up of devoted scientists, many of whom are experts in their field, that try to do one thing: try to understand what the truth is of the science that you're doing, and understand and ensure the safety for the population of the US. That
Mm-hmm.
is their goal. There's nothing more. And I've always had good experiences because I was able to move in directions that I might not have moved in if I was just thinking about it myself. So this is a critical agency. We've had discussions with them. They knew our intention. They gave input into the protocol. We agreed to conduct a very large preclinical toxicology package, which is necessary to make sure that there's safety. And there is, and we've done that.
Mm-hmm.
And we now are at a stage where we've created a state-of-the-art protocol that has started. There are going to be 25 sites across the US and Canada, some of the best ALS sites really in the country and the world. It's going to be 120 patients. It's a
Mm-hmm.
placebo-controlled trial with 2 treatment arms, one of them given the same dose given to those patients twice a month, and the other one, the same dose, but only given once per month, because we think that could be an effective dose as well. And then there's going to be a placebo group. And the endpoints are going to be the difference in the ALS FRS at 6 months. So after 6 months, we'll have top-line data and know whether this is a drug that is highly effective and safe, and then we would move forward. In addition, there's going to be a 6-month extension. That's a
Okay.
blinded extension. So
Mm-hmm.
people who are in the group that are, and are responding, they can continue to move forward for a full year in a blinded manner on the dose that they're on. And the placebo group has a chance now to be re-randomized into one of the 2 therapy groups for the remainder of the study at the end of the year. But we plan based on that data to submit that data. And if the data are good, then we're on our way to perhaps getting a potential approval for a therapy that can make a difference. And I want to talk about that for a moment as well.
Sure.
People with ALS really suffer through this disease, unfortunately, because there are very few and really no adequate treatments at this point. Unfortunately, when someone is diagnosed with ALS, they will often die between 2 and 5 years.
Yep.
And the progression, as I mentioned before, is significant, and people become dependent on devices depending on what area of the motor neurons are destroyed in the disease. So it can start with a calf muscle coordination. It can start with an arm. It can start with the ability to swallow. And unfortunately, each month, this can progress, and people become more and more dependent on devices just to live, whether it's a motorized chair. Sometimes they're left with only the ability to move a finger.
Mm-hmm.
They have devices set up so that they can communicate through their eye movements, help them get nutrition, and they become dependent on caretakers and family. So there's such an unmet medical need in this area. And that's one of the reasons that we're moving really rapidly to complete this study and move forward with this potential therapy. The other thing I should mention is that the management team and people who are associated with this company have either they themselves either experienced this disease through their family or have the gene. And there's one person who's associated with our company, Daniel Barvin, who's been in the media, who has the gene that
Yeah.
makes him susceptible for possibly getting ALS or frontotemporal dementia. So we're moving forward in ALS, but also this combination is agnostic to
Right.
neurodegenerative diseases because it's such a clear, basic mechanism. We're not going for this specific target in some downstream mechanism. We're lowering and tamping down inflammation to stop progression, whether it's in ALS, frontotemporal dementia, Alzheimer's, Parkinson's. So we are moving forward, and we'll be submitting an IND in frontotemporal dementia in the coming weeks, because we plan to study that as well, since both frontotemporal dementia and ALS share a lot, both genetically and sometimes the two go together. So there are a lot of similarities in terms of inflammation, regulatory T cells. So we think that there's potential for this therapy in frontotemporal dementia, for which there are no treatments at all. And then from there, we'll be moving in other directions as well that we haven't yet disclosed. So this is sort of a pipeline within a singular product, but the basis of Coya is combination therapy to affect the immune system. And so this is one of other types of combination in our pipeline. Well, one such combination, which is Coya 303, is combining GLP-1 with low-dose IL-2. And the reason we're doing that is that there is evidence that people who have been taking GLP-1 for diabetes may have had or may have a lower incidence of Alzheimer's.
Yeah.
And Novo Nordisk is about to release a substantial amount of data in 2 trials studying GLP-1 in large cohorts of Alzheimer patients. So that's going to be presented next month at a scientific meeting. We believe that GLP-1 alone isn't enough to have a significant impact, and it needs that combination with low-dose IL-2, which will affect regulatory T cells. So we've done some work with this combination. We've done some preclinical work in the lab to show that this combination is not only additive, but synergistic in tamping down inflammation. So we've shown in the lab that this combination has potential. We then moved into an in vivo model, an animal model, and we haven't fully released that yet, but I can say that we're seeing some interesting findings in how it's reducing inflammation. So when Novo Nordisk releases their information, if it's equivocal, which it may or may not be, but if it's equivocal, then we're going to have some direct line of sight in working with large pharmaceutical companies in terms of this combination in Alzheimer's disease, because a company of our size cannot do Alzheimer's disease on our own, unlike ALS and frontotemporal dementia. So, sort of in full circle, at a time when I was thinking of moving out of the industry and retiring, this pulled me back in, because it's almost a no-brainer. Treating rare diseases that have unmet medical need, a parsimoniously explained mechanism of action that's simple, that's clear to understand, biologics that affect that system directly, working with people one of whom wins the Nobel Prize for Medicine, and Dr. Stan Appel, who's a giant in the field. It's almost a non-decision. It's obvious that if one has an interest in doing something important that can bring therapy to these patients in need, this is a good place to be.
Yeah, and it's funny how our industry has that effect on us, always dragging us back into the next greatest thing. But yeah, I mean, what you're describing here, Fred, it's a combination drug story, it's a repurposing story, it's immunoregulation story. It's extremely elegant in the sense, obviously you're starting with rarer things, but how this trickles to Alzheimer's and Parkinson's and everything else, from the initial work on ALS and frontotemporal dementia. Yeah, I mean, it's a beautiful story, all wrapped up, and I'm excited about it. Could you say? I know it's early running, and obviously drug companies and biotech companies can only do so much, but you do, and then we chatted a little about your cell therapy experience. Can you just say a few words while we have you about the sort of the cell therapy part of this? I know that's a little early on, but you are thinking again about expanded T-reg products as well as T-reg derived exosomes, which in the cell product area is kind of hot as well as a topic. Could you say just a few words about your thinking there as
Sure.
well? Because,
So we've made a turn to combination therapies from regulatory T cell therapy. However,
right.
we are exploring exosomes as well. And exosomes are very interesting. Exosomes are the transport system of the body, and they go everywhere. They don't get inhibited by the blood-brain barrier. They can flow anywhere. So there's a lot of interest in exosomes. And there are two areas of interest for us. One, if we can, and we've been doing some work on this,
Mm-hmm.
extract exosomes from regulatory T cells, they will have the same functionality as those cells, meaning that these packets can actually reduce inflammation.
Mm-hmm.
And so one's imagination can go very extensively wild in this concept because the exosomes go unimpeded throughout the
Mm-hmm.
body. So you can think of a whole variety of diseases that this could possibly affect by reducing inflammation. The other thing is that they're relatively easy to create an allogeneic product from it so that
Mm-hmm.
you don't have to use the patient's own cells. You can have donors and create a
Yep.
large bank of exosomes, similar to what Mesoblast does
Yep.
with stem cells. So there's a lot of reason and a lot of interest in that. The other thing that we've done, we've partnered with Carnegie Mellon and have done some experiments that were interesting, which attached certain proteins to the exosomes. So not
Mm-hmm.
only are we thinking about exosomes in and of themselves as a potential therapy, but using them as a vehicle with specific proteins that can pinpoint areas of the body. So this is a new emerging and exciting area for us. And as you mentioned, though, as a small company, we have to first focus on what's in front of us. And what's before us is to really focus on ALS,
Mm-hmm.
FTD, and continue to move forward, as well as this combination with GLP-1 and low dose IL-2. So
Mm-hmm.
a lot of excitement now within the company.
Yeah, I can imagine. And again, it's one of those things, and we talked about the potential of starting with one indication and trickling down to so many other areas. We talked about the pipeline, obviously a lot going on there. What else should we be on the lookout for in terms of the company as we get into 2026? Other public facing initiatives, conferences, talks, anything else while we have you today, Fred? But it's a really exciting story.
Sure. Well, we are aligned with NEALS,
Okay.
NEALS, which is really a prominent organization of investigators that's patient-oriented
Mhm.
and make sure that there's availability to patients and investigators with the latest thinking in science. So we're aligned with that group as well. Moving forward, I think things to look at now are there's going to be more data in FTD with this combination that's being generated at Houston Methodist. There was some already that has been shared that showed that this combination is having an effect on cognition, a positive effect,
Mhm.
as well as reducing inflammation. So there'll be more data released this year in that regard. There's also more data that was released initially in an Alzheimer's study with just low dose IL-2, and there'll be more follow-up to that as well. And we will be releasing and hopefully publishing the data on the combination of GLP-1 and low-dose IL-2. And of course, with respect to ALS, we'll be reporting how the study is moving along. And once we get to the top line data, of course, we'll report that, because that's going to be critical data not only for the medical scientific community, but also to allow us to move forward in submitting a potential application for approval. We will also be initiating the FTD study, so that's going to occur as well. And of course, we'll keep everyone posted with other kinds of meetings where we might be presenting or having discussions. Also importantly, let's not lose sight of the fact that at the center of all of this is patients. So we will be participating in a lot of patient-oriented advocacy to continue to keep that engagement going. And then some of the lesser things, such as walkathons and marathons, although I may set out a couple of those marathons. But yes, we'll be reporting on all of those activities as we move along.
Awesome. Yeah, it's a very exciting portfolio. Obviously, it's T-regs with the recent Nobel Prize news has been at the forefront, and it's exciting to see how quickly you're moving with this and getting things closer to patients and into patients and getting top line data. But I'm rooting you on. I'm excited for you. Not that everything you hadn't done before this wasn't exciting, but this is extremely exciting. So, obviously wishing you and Dr. Appel and the team the best with all of it as you continue to go down this path. Again, for everybody out there that's going to be listening to this episode across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Fred Grossman, President, Chief Medical Officer, Coya Therapeutics, doing really amazing work to develop these multi-modality T-regulatory therapies for a range of neurodegenerative diseases, as well as autoimmune and metabolic disorders. Fred, I want to thank you for taking the time to walk through this really exciting story with us. Obviously, thank you for everything you do for patients. And as we like to say on our show, thank you for creating a better tomorrow for literally millions of people out there via what you're doing. Really great story.
Thank you, Ira. Great to be here.
Look forward to continue to follow you.